This is the third part of the chapter on linkage, chapter 7. So last time we talked about how recombination mapping can be used to make chromosome maps and to place genes on the chromosomes in the correct order. Now, I cannot overemphasize how very important gene mapping by recombination was historically. You will still see many chromosome maps with the units of centimorgans on them, and those results still help geneticists in their work today. However, most people aren't doing that anymore, and so I think it's important for genetic students to understand this historical aspect, but today DNA sequencing has gotten so cheap that people just go right to that rather than looking to make a linkage map. I would like to go in this chapter and talk about how linkage is used in some other very important ways. For many years, linkage was actually the only way to figure out the genetic basis for a certain trait or a phenotype on the trait level. So we're going to talk about how linkage is used to determine the genetic basis of these traits and also to predict whether someone might be a carrier for a disease allele. So we're going to start off by looking at an example where you can follow one trait by the inheritance of another trait. One example is a autosomal dominant disorder called nail patella syndrome. And so this syndrome is you have very poorly developed fingernails and toenails, and you actually do not have a kneecap or a patella at all. So it turns out that this dominant disorder is actually linked to the gene that encodes the blood type, the ABO blood type. And so the idea here is that you have a recombination map and you have a physical map. So we're going to start off with the recombination map. You look at these traits and you can see in a family that the nail patella syndrome tends to be inherited with an allele of the ABO blood type. So those are things at the trait level that we would put on a recombination map with the units of centimorgans or map units between them. Now there's also a physical map, and if we already know the location of the ABO blood type gene on the physical map, what that's going to help us to do is to locate the general area of the chromosome where the nail patella gene lies. And so that's how a couple of disease genes have been discovered. And in fact, now we know on the physical map as well as the recombination map that the nail patella gene is right nearby to the ABO blood type gene. The other useful thing is that the ABO blood type can actually be used as a proxy to predict whether you have inherited the disease allele for the nail patella syndrome. So since they are so close together on the chromosome, it's pretty likely that if you get one thing, you're also going to get the other one that is linked to it. Now we're going to take a look at pedigrees with linkage, and we'll go into a lot more detail in class. But what I'd like to point out here is that we have indicated both nail patella syndrome. So if you are red, you have nail patella syndrome. And since it's dominantly inherited, roughly 50% of the progeny will be affected. What we also have on this pedigree is the blood type, and there are 3 different alleles for the blood type gene. There's the 2 dominant alleles, they're actually co-dominant, which we'll talk about soon in the class. So big IA and big IB, and then there's also a recessive allele, little I. These can join up in different configurations, and if you have 2 recessive alleles, you're going to be blood type O. If you have either 2 B alleles or a B and a little I, you're going to be blood type B. And if you have 2 A alleles or an A and an I allele, you're going to be blood type A. Incidentally, there's also blood type AB, and it just so happens that there's no individuals with that blood type on this pedigree. So if you look, what you can see is that individuals got one chromosome from one parent, one chromosome from the other parent. And so if we look at this individual, this first son right here, what we can see is that he has type B blood. And so the only way you can have type B blood is if he got that B allele from his mother and the little I from his dad. So that will actually predict the other things that came with it, right? So if he got the IB from his mom, he probably got little n, which is the normal allele for nail patella. If he got little I from his dad, then he probably got the disease allele for nail patella. And in fact, he shows the disease. Now, I'd just like to point out that sometimes you can get recombination. And so sometimes the ABO blood type gene and the nail patella gene will not be inherited together because a recombination happened between those two genes. So we can actually figure out individuals that are recombinant based on the alleles that they have. And this is something we'll actually do in class, is we'll use that same pedigree to see which ones are recombinant. Now, we can not only locate ones that are recombinant, we can figure out, well, how many are recombinant out of the total individuals, right? So out of the total that we know for sure whether they're recombinant or parental type, what percent are recombinant, we can actually calculate that recombination frequency, and we can predict the centimorgan distance between those two genes from a pedigree or combining data from multiple pedigrees. This is something we will be doing in class. Okay, I'd like to move on, however, and point out that this is a really unusual situation where we found two traits that were linked together. Most disease genes, we don't have an obvious observable trait that we can use to follow the inheritance of that disease allele. And so what people have moved toward doing is using something called molecular markers rather than an actual trait to follow the linkage of a particular gene. And the idea here is that although you cannot observe a molecular marker by looking at a person, you can't know their phenotype, it's very easy to actually get some DNA, collect some blood, do a DNA test, and figure out which form of a molecular marker that person carries. So let's just talk a little bit about what a molecular marker is. So it is a position in the genome that tends to be polymorphic among individuals. So surprisingly, humans actually share around 99.9% of their DNA. So if you look at any two individuals, they're really virtually identical at the DNA level. Of course, that 0.1% that we have different from each other is of great interest. And so those differences are what leads to the phenotypic difference among individuals. Some of those differences might explain why my family has a high incidence of breast cancer and yours doesn't, for example. Other changes at the DNA level are silent changes, and so these changes may happen in a non-gene region of the genome, and there's really no phenotypic consequence from that difference. So if we look at that very small amount, that 0.1% that is different, it sounds like this negligible amount of DNA sequence that actually varies between individuals, but in reality, it is actually an average of one difference per 1,000 bases of DNA. And we've got a lot of DNA. So humans have 3 billion base pairs, roughly, of nuclear DNA. And so if we have a difference every approximately 1,000 nucleotides, that means we've got 3 million differences, roughly, between individuals. And so there are a lot of possible places where we could use in the genome to find a molecular marker that can be used to follow the inheritance of a nearby disease allele. So most variation between individuals falls into two categories. So either those are SNPs. SNP stands for single nucleotide polymorphisms. The other kind is something called VNTRs. You'll find a number of different names for these, but the idea of this is that it's a variable number of repeats. So let's just use this particular figure to illustrate what a SNP is versus a VNTR. So what you see here is a very short region of a genome, and it's only 35 bases, all right, and you have 7 individuals sequences and listed here. So what you see is that in some positions, we've got the same exact base between individuals. So in position 1, everyone has a G. In position 4, everyone has an A, etc. There are some places where people tend to vary. So in position 2, for example, it's very likely that you either have a G or a C, right? It looks like roughly 50-50 in this very small population. There are a number of SNP sites. Here's another one at position 9, another one at position 5. And from this very small sample, it looks like the one at position 9 and the one at position 2 are much more polymorphic in the population than the one at position 5. So position 5, only one individual out of 7 has a different base. So for SNPs, there are really only 4 bases that are possible, so we call these different forms of the SNP. And so another term for that, for different form, is a different morph. We can detect what kind of SNP a particular person has at a certain location by either sequencing their DNA. We also, very commonly, we use something called a microarray, which we'll talk about later in the course. And historically, people used to use something called restriction fragment length polymorphisms. For VNTRs, here's an example down here at position 25. So you see that there's a repetitive region in this sequence. There's an AG, AG, AG. And what you see here is that 3 of the individuals have additional repeats of the AG, whereas 3 of them, they did not have those additional 2 repeats, and so we just stretched out the sequence there so that everything else would line up. All right, so these guys only have 3 repeats, and these ones up here have 5 repeats. The nice thing about VNTRs is that many different morphs are possible. You're not limited to just 4 possibilities like you are for a SNP. Just want to say a little bit more about VNTRs. So these fall into 2 classes. The one that we previously saw was a microsatellite DNA marker, and this is because it was a very short sequence that was repeated, only 2 bases, right? So a microsatellite is defined as 2 to 5 bases that are repeated, whereas mini satellites have longer repeats, so maybe 15 to 100 bases that are found in several copies. So the reason why VNTRs tend to be very polymorphic in the population is because of replication errors during S phase when a cell is dividing. And we'll talk more about that when we talk about the process of replication. Finally, VNTRs are detected by a technique called polymerase chain reaction. We will again talk about this later in the course, but the idea of this is that you use synthetic primers, small pieces of DNA, which will kind of focus in on the region of the genome that you're interested in, the one that has the different number of repeats, and you have a set of 2 different primers, and this technique, polymerase chain reaction, will make many copies of the DNA between those 2 primers. And so you end up with lots of copies shown in red here, and the size of those copies is going to tell you how many repeats were found between those 2 primers. So for example, if you have an individual with only 5 repeats, that's going to be a smaller band on a agarose gel than an individual with 9 repeats, which would be a bigger band. Some people will be homozygous for the smaller size, some people will be homozygous for the larger size, and some people have 1 allele of each of those. VNTRs are also what is used in forensic DNA analysis. So 13 different microsatellite loci are tested. So the number of repeats that the suspect has is compared to the number of repeats that is found in the DNA from the crime scene. This is also how paternity testing is routinely done. So just to end up, molecular markers are used in this very same way that we would use that ABO blood type gene to predict whether Neil Patella syndrome has been inherited as well or not. So We just get DNA from an individual and measure what form of the molecular marker is present. And the individual has to be heterozygous. So for that molecular marker, that individual has to have two different morphs of that marker in order to be able to follow whether a disease allele was passed on. And just as a summary, we can use molecular markers when we are initially looking for the actual gene that is causing a disease. We can use it to calculate recombination frequency to figure out how far away from a molecular marker that disease gene might be. Or we can also use it to predict whether an individual inherited a disease allele.